Okay. Nampak tak?
Nampak.
Nampak Okey, jap. Saya mana pula dia butang ni? Okey. Alright. So Assalamualaikum. Very good morning, everybody. Thank you again for making the class this morning. I'm very sorry that I actually had to revert to a online class today because I had something going on. So saya rehat sebentar daripada perkara itu dan kita try and make a class lah. Just to apa tu, Danu nak record lecture, tak apa. Okay, what we're gonna do is I'm going to put up a recorded lecture later on Putra Blast. So nanti korang boleh tengoklah the video. I think after this I'll do it. Before we move on, kepada sesiapa yang telah menghantar ataupun sedang ingin menghantar practical report, don't worry about it. Kira bagus juga lah korang ni actually nak menghantar on time. But I will put up the submission because now I need to know your grouping first from Dr. Fiza. I'll get the grouping from her and then I'll put the submission page on Putra Blast. So don't worry. I'll probably open the submission hopefully by tomorrow lah, last day of the week kan. So you can start submitting. Right. Everyone okay? I assume semua orang kat rumah and knowing that it's a morning class, mesti korang semua either belum mandi ataupun baru bangun. Yes.
Can we extend the deadline for the practical because my girl did some mistakes and then tomorrow we have exam, so can we plan to amend it after the exam.
So if that's the case, I will leave the submission page open and I'll extend the deadline to next week lah. Is it okay? Okay, all right. I think some of you messaged me yesterday night, asking about using sequences from non-redundant databases and our database kan, whether it's good or bad. The short answer to that is there's nothing wrong with using databases from NR databases, simply because sequences from non-redundant databases may or may not be correct. But provided that those sequences are valid but waiting confirmation, it's okay to include in your practical. But bear in mind that sometimes when you do an MSA, the sequence ataupun the arrangement of the sequences redundant may contain or may not contain certain conserved residues that are confirmed. Kan, but it's fine, you can use non-redundant, there's no problem at all. Okay? Right. Okay, sekejap Okay, give me 2 minutes. All right. Sorry about that. Okay, so yes, you can use the results from NR, but since you have time now, you can actually redo it if you want. But if you want to remain the sequences from NR databases, it's fine. It just means that the sequences that you get from the NR database may or may not be valid, but it's still okay for the purpose of the practical. Okay, right. Okay. Before we go ahead with the class today, how was test 2 last week? Okay ke?
Boleh lah.
Boleh lah Okay. So it's a bit better. I don't want to say better lah, but is the questions bearable ataupun susah betul? Ataupun lagi susah? Macam lompat keluar daripada mulut singa ke dalam mulut buaya macam tu.
Bearable kot sebab satu slide je, satu topik je kan.
Yeah. Results. Okay. I haven't marked your. I've only marked a few scripts, and then it seems macam okay lah. Yeah, I also think it might be better than test one. Tapi nanti kita tengok. Hopefully you guys are gonna do much better than test two. Right. Okay. So let's begin. So the idea for this morning's class is for me to make it as brief as possible. We're probably going to have only a 1 hour, 1 hour and a half lecture to this morning, where we are going to go into the second last topic for the course, which is molecular phylogeny and evolution. Okay. So in this particular topic, what we're going to look at is actually we're going to look at how you can use sequence data or the sequence that you take from a particular set. That means it's the same with MSA, where you select sequences, be it from any database, can be non-redundant, it can be ref sequence, and then you use information that is contained by those sequences to build a phylogenetic tree. What that means is, instead of looking for similarities or shared residues or shared characteristics between those sequences, what you now use is you use the whole information that you have to infer relationships. That means how far along they are, or the relatedness between those two sequences, right? Let's say, for instance, you pull in sequences or protein sequences belonging to, let's say, protein orthologs. As for example, maybe you ambil globin orthologs kan, from human beings, from cats, or from whatever other types of organisms, and then you use the sequence, the data in those protein sequence to determine how are those organisms, right? So I say this. I actually put emphasis on this because if you look at all of the topics that we've learned from the beginning of the course until MSA, what we are focusing on is we're looking at similarities, right? We're looking at similarities, and then we use those similarities to actually make a score. But in molecular phylogeny, what we're doing is we are now focusing on what makes them different, so that we can actually infer relationship, or we can infer some sort of relatedness in terms of distance. So in molecular phylogeny, what we are focusing on is actually on the differences, whereas for all the other topics, we mainly look at similarities. Okay, so far so good. Okay, right. So now the LOs for this topic are as follows. You can read this later on. You can go through it in your own time. But in this lecture, I'll introduce to you the molecular clock hypothesis, as well as the nomenclature of phylogenetic trees. And maybe next week in face-to-face classes, we'll probably talk about the 5 stages of how we can build or how a program generates a phylogenetic tree, right? If you look at the costs, right? Everything that a program does goes through a lot of steps. There's a lot of steps. If you look at BLAST, it's a 3-step process: scan, hit, extend, right? If you look at SciBLAST, there's 4. So that's where you repeat the BLAST over and over again, and there's actually building the possums and etc. If you look at MSAs, there are 5 approaches to building an MSA. So there's a lot of those numbers going on step by step. So in molecular phylogeny, a program such as MEGA also utilizes some stages, and it's actually 5 stages. We'll look at that next week. Okay. So far so good. Or should I rewind, or should I go slower? We try and we try and finish by, I think, 10:30 lah. We try. There's not a lot of information I want to actually share this morning. We'll try and make it brief. The reason why I'm trying to break down the topic is because this topic is quite dry. So I'll have to apologize in advance. It's dia kurang menarik compared to MSA. Macam MSA tu menarik, tapi it's less menarik than MSA. So you might feel a bit macam dry lah. So we try and make it. We try and do it as slow as possible. We break it into parts. So let's look at first and foremost, what is actually molecular phylogeny or molecular evolution. When we talk about evolution, it's obviously a process of adapting to changes. Kalau kita, if we take an organism, for example, let's say the human being, right? So a human being from our, ancestors, be it who they are, they have gone through a lot of adaptations to, to survive, right? And because the environment always changes. So at the physiological aspect of a human being, there's a lot of things that are now new in us that were not possible in our ancestors, right? And all these sifat-sifat, all these characteristics that are manifested in us comes through changes at the molecular level, which means there are changes at the DNA level, or even at the protein level, or even at the expression of a particular protein, right? So the study of the changes on those sequences, be it gene sequences or protein structure changes, in an organism or between organisms is what we call molecular evolution, right? So to summarize this particular definition, everything that has to do with studying the differences between sequences that leads towards a process of evolution is called molecular evolution. Boleh? Okay, right. So that's molecular evolution. Molecular evolution refers to a process. Phylogeny, on the other hand, is the ability for us to determine relationship or to the two sequences or two proteins are very close together or distantly together by virtue of a particular scale. It can be distance, it can be changes in amino acid residues. That's called phylogeny. Okay. For instance, we know that there is a difference between us and, let's say, chimpanzees, obviously because we don't look the same, right? Because that means there are characteristics in chimpanzees that are different than ours, but there are also some similarities that we have between ourselves and the chimpanzees. But we use information that we have between these two organisms to determine how far apart we are. So that's called phylogeny, right? So if molecular evolution is the use of sequence data or the study of changes in sequence data, be it gene or protein, and phylogeny means the ability to determine relationship. So molecular phylogeny is the use of molecular information or studies in the changes of genes and proteins to determine relationship between multiple organisms. So that's molecular phylogeny. Okay. So far so good, ada confused. Okay, diam tanda setuju ataupun diam tanda faham ataupun diam tanda tertidur lah, either one. Right, I repeat. So molecular evolution is the study in which you look at changes or differences between at the gene or protein sequences between many types of proteins or genes, right? Phylogeny is the ability to determine relationship, whether they are closely related or distantly related, or where they are associated in a family. Molecular phylogeny is the use of gene and protein sequence data to determine relationship between sequences. All right. Okay. Right. Now, so when we talk about molecular evolution, we talk about molecular phylogeny, relationship and relatedness between sequences. The whole concept of this actually came from Charles Darwin in 1859. We assume, let's just assume, irregardless of religious belief, let's assume that Charles Darwin has some dia betul lah. That means correct in the sense that there is a theory called the theory of evolution. According to Charles Darwin, he said that there is a natural process in which he calls or terms it natural selection, where nature will select for the species which is superior to its predecessor. Ataupun in simpler terms, there is always a process of mutation in an organism. Because of changes in the environment, then an organism will change to adapt to that environment. According to Charles Darwin, the inability of a particular organism to adapt or mutate to change will lead towards its extinction and the succession of a new organism. Contohnya, for instance, the ancestor of a human being and a chimpanzee more or less mutated or evolved to diverge into two species, or it became extinct and it led towards the birth of two new species. Can be of two things lah. Okay, so that's in terms of of organisms. When you look at it at protein levels, you can actually now understand that there are certain proteins that are available in one organism but is not available in another organism. For instance, an extreme case will be proteins that are present in, let's say, fungi, which is not available in, let's say, bacteria. And because they are connected by one ancestor, that ancestor may have evolved to diverge into the species of fungi and bacteria when it adapted to an environment. So the proteins that lead towards its survival will be selectively expressed in either one of those organisms. So far so good? Macam sedikit complicated, but hopefully korang understand lah. Yeah. Okay. Is that okay? Can I go ahead? Boleh eh? Okay,
Boleh,
boleh.
Doktor.
All right. So if you look at the slide over here, you can see three types of animals. Kan? The first one is the koipu, the second one is the thylacine, the third one is the platypus. If you sort of recall, I don't know whether you've ever seen a koipu or if you've ever seen a thylacine. I'm sure some of you, if not all, have knows about the platypus and maybe have seen one somewhere like at zoo, kat mana-mana kan. It depends. But if you look at these three animals or organisms, they are actually at the juncture of the process of evolution or the process of diverging species. If you look at the koipu, physiologically or on the outside, it looks like the combination of a zebra and a donkey, right? I don't know kenapa dia tak panggil zonky je, but dia panggil koipu, tak kisahlah. And if you look at the thylacine, it looks like it's actually a mix between maybe a tiger and a dog, kan? Or maybe a fox, or maybe another type of feline species, kan? So the koipu and the thylacine are actually organisms that occur or exist before the deviation of the donkey and zebra, as well as the tiger and the let's say dog in this case. So it is thought that when the koipu and thylacine evolved, it led towards the extinction of koipu and thylacine species, but it led towards the divergence of two new species, which actually this is the zebra and the donkey, as well as the tiger and the dog, right? If you look at the platypus, on the other hand, it's still here. But if you look at the characteristics of a platypus, it sort of looks like a duck, tapi macam bukan duck. Nampak macam memerang, tapi macam bukan memerang. It looks like a guinea pig, tapi tak ada bulu, right? But in the inside, genetically, a platypus, or physiologically as well, a platypus is one of those mammals which has characteristics of a reptilia, which means it, or amphibian that method, where it actually lays eggs. So it is, we can now start to understand that the platypus might be or might come from an ancestor which was the deviation of either an amphibian or a mammal, which actually evolved to merge together. So that means evolution can go both ways. It can start toward the deviation ataupun gabungan. I don't know what gabungan. The pairing of two new organisms. Okay, right. So those are actually animals that we can actually see, benda yang we can actually tangible, and you can actually imagine. So let's now move our perspective into looking at protein sequences, right? So if we understand that when an organism starts to evolve, mutations will occur. So in an animal or organism, you can see it from the outside because it's a phenotype that comes from a genotype, right? But now think about a protein sequence. Let's say, for instance, here we have the sequence of insulin. Okay, so when I think insulin, actually the protein insulin has been sequenced in the early 1960s, but the protein has been discovered much earlier. So when they actually did the sequence analysis, right, they found out that the insulin protein sequence can be categorized into four domains, right? The first one is the signal peptide. The second one is the B chain. The third one is the C peptide, and the fourth one is the A chain. The signal peptide occurs at the beginning or the end terminal of the protein, and you have the A and B chain being apa namanya diasingkan, separated by the C peptide. Okay, what's interesting is that when they did a molecular multiple sequence alignment between insulin sequences or orthologs, which means insulin from cow, sheep, sampai like a chicken though, they found out that they can actually align those domains together: signal peptide, B chain, C peptide, A chain. It's never a different sequence. It's never signal peptide, A chain, B chain, and C peptide in certain animals. They're all the same. What they also found out was that the B chain is actually separated from the C peptide by an arginine or RR sequence, while the C peptide is almost always separated from the A chain with a KR sequence. K apa? K arginine code. No, R is arginine. K maybe lysine code. Then check. But that means there is a bridge that connects between the B chain and C peptide as well as the C peptide and A chain, which is actually a conserved motif, RR and KR. Okay. What they also found out that when they actually looked at the MSA between all those orthologs, they found out there are almost no changes in the B chain and A chain. As a matter of fact, if you look at the B chain, it's almost 100% conserved from all the species. We're not even talking about species that live in the same habitat. These are like, in cows, and then in sperm whales, which are in different habitats and different types of organisms. One is one is large, one is smaller. One can be herbivore and omnivore, right? In the A chain, on the other hand, what they found out was that there is some deviation or differences, but it's very specific. That means it only occurs in a 3 residue area kat tengah-tengah A chain, right? If you look at the red box over here, that's the only variable sequence in the A chain sequence of insulin from all the orthologs. Everything else is almost 100% conserved. Further analysis of those particular 3 residue chains, they found out was that the changes were actually conservative, which means even if there are any mutations, it's with amino acids from the same biophysical property. What the difference comes in the form of chains. So certain insulins, they bind, they have different chains, 1 or 2 chains, disulfide loops, disulfide chains. So the loops are different, but the folding is almost the same. Okay, so tak apa. So what's important is that there are almost no significant changes between the A chain and B chain. So if you remember from MSA, from your practical kan, I've mentioned that when we have a conserved sequence or conserved domain between a lot of orthologs, it sort of tells us that that particular area is actually very, very important. And that area relates towards the function of the particular protein. So from here, they actually sort of surmise that the A chain and B chain is related towards the function of an insulin and cannot be changed drastically. Kan? Conversely, when you look at the signal peptide and the C peptide between the A and B chain, they found out that it's highly variable, right? If you look at the signal peptide in most of the orthologs, you can see there's a lot of conserved areas there. But when you compare between the sperm whale and elephant, there's almost no signal peptide available, right? Whereas if you look at the C peptide as well, between those sequences, you also have a lot of variable regions. We have a lot of gaps between the cow and sheep and human insulin, pig and dog insulin. And in sperm whale and elephant, there's no C peptide at all. It sort of tells you that the B chain is directly connected to the A chain, right? So what they found out was that certain areas inside the sequence changes by the process of evolution. And the changes that occur at those areas are often, it doesn't actually lead towards the protein losing its function. You know what I mean? what I mean to say is that if you look at the differences in the insulin protein between the orthologs, the changes, a lot of changes only occur in the signal peptide and the C peptide, which means if there are any mutations or changes in amino acid, it rarely happens in the B chain and A chain, but it happens at the signal peptide and the C peptide only, right? And what they also found out was that when they use histological data or paleontological data, that means they look at fossil data, uh, they actually found out that the changes that occur at every single area doesn't occur randomly, which means that it doesn't change 1 year and then in 1,000 years it changes, and another 500 years it changes. There's actually a constant time in which a residue will mutate. What they found out was that the rate of mutation or changes inside a particular domain, in the B chain, for instance, one amino acid change only occurs every 10 billion years. And the same is for the A chain. The change in one amino acid only occurs once every 10 billion years. If you look at the changes in the C peptide and the signal peptide, they found out that one mutation occurs once every 1 billion years. Ni nombor ini 0.1 times 10 power 9, 1 times 10 power 9. So that means the amount of amino acid residue that change. Okay. You don't have to worry about this. You can just read it. This just tells you why there is a change in there is a amino acid change in in human insulin, guinea pig insulin, mouse insulin. It just tells you that some mutations are there because it wants to adapt to a certain environment. But leading towards that this slide over here, where I mentioned that the amount of change at a particular area occurs at a constant rate and it's not random, leads us to one hypothesis that is actually coined by Linus Pauling and E. Margoliouth, right? It's called a hypothesis because there is no way for anyone to confirm that this is actually true. I remember in science you have a few nomenclatures. You have theory. That means you have a scientific hypothesis that has been proven either by calculation or by scientific data. Then you have law, which means it's a scientific hypothesis that has been confirmed through scientific experiments and occurs in nature almost naturally. And then you have hypothesis. So theories can be theory of relativity, kan? So the theory of relativity was coined by Albert Einstein, and it has been proven by calculation. But unless you can, run at 2 times speed of light, you can't really prove it in terms of scientific terms. But the work at the Hadron Collider is actually trying to make this theory a law lah. Then you have law. Law is what? Law of gravity, right? So law of gravity, Isaac Newton coined that. Law of gravity has been confirmed by calculation and experimentation, and it occurs every day, and it keeps us all on Earth. And then you have hypothesis where it's a scientific hypothesis which has not been confirmed through scientific data because no one lives that long, and no calculations that can actually prove that the molecular clock actually works or not. But it is a hypothesis which may or may not be correct lah. But according to Linus Pauling and Emmanuel Margoliouth, they state that the molecular clock is a type of property found in certain species, for instance, sequences or protein sequences, where for every given protein, the rate of molecular evolution. So remember, molecular evolution means changes in sequence, protein sequence or DNA sequence, sequence punya composition is constant in all dia punya evolution process, which means that, okay, I'll go back. If you look at the insulin sequence over here, there is a molecular clock to insulin, which tells you how much mutation occurs during a particular time frame between insulins from cow until chicken. That means from the ancestor sampai all of these organisms. All right? Okay. Everyone okay? Confused ataupun nak saya repeat? So far so good, I assume everyone so far so good. Hopefully tak ada so far so good. Bye. Okay, I'm going to skip this. I'm going to skip this equation over here, but I'll try and explain the paragraph here. Okay. So in 1971, Richard Dickerson, what he did was he took three types of classical proteins that are found in almost all organisms, and has a lot of sequence data from early organisms to recent organisms. Okay, cytochrome C, hemoglobin, and the fibronopeptide protein. What he did was he merged historical data, which means the first occurrence of that species until the recent one. And then he looked at the change in amino acid. That means he did an MSA, and he looked at a lot of the changes that occurred. Okay. From there, he actually made a time plot, right? And the plot, okay, that means how many amino acid changes happen per 100 residues. So let's say if let's say every 100 residues, how many changes occurred in a particular protein sequence. Then they plot lah. What happens was that when he plot that changes towards the historical data, and then he tried to actually buat garis apa to line it, to draw a line. He got an almost straight line. If you can see for cytochrome C, it's actually a straight line lah. For hemoglobin, it's almost a straight line, and for fibronopeptide, it's actually a straight line. This tells him that for a line to be straight or to be linear, the changes actually occurred at a constant rate. That means there is a design towards the mutation of that particular protein. What he found out was that for hemoglobin, the molecular clock for hemoglobin is 5.8 million years. For fibronopeptide, it was 1.1 million years. For cytochrome C, it was 20 million years. Right. So the first thing to look at here is if you look the lower the molecular clock, which means the lower the number here, the more slanted or the more bukan cekung, apa nama dia? The more cerun apa bahasa English? The more slanted lah. It's sharper. The line is sharper. The lower the number is. Steep, yes. Thanks, Daniel. So the steeper the line is, the lower the lower the number, the steeper the line is. The second thing to understand is that the molecular clock over here tells you how frequent the amino acid change per 100 residues occurs. So for hemoglobin, the molecular clock for hemoglobin is 5.8 million years. That means for one mutation or for one change to occur per 100 residues in hemoglobin, it takes 5.8 million years. Or every 5.8 million years, one residue per 100 one amino acid per 100 residue in hemoglobin changes. Does that make sense? So in cytochrome C, because the line is less steep and the molecular clock is 20 million years, that means every 20 million years, there is one mutation in every 100 residues of cytochrome C. Okay. Okay, tak? In fibrinopeptides, one amino acid change per 100 residues occurs every 1.1 million years. That's what the molecular clock says. Okay. If you look at the table over here, there are also different types of proteins, and there are also numbers on the right side. The numbers on the right side tell you the molecular clock for each of those proteins. For instance, I'm guessing this is actually fibrinopeptides, gamma fibrinopeptides. This is a different fibrinopeptide. So the molecular clock is 9, kappa casein is 3.3. But these numbers mean 9 times 10 power of 9, which means let's say lysozyme, right? Lysozyme has a molecular clock of 0.98, or let's say almost close to 1. That means one mutation in lysozyme will occur every 10 billion, every 1 billion years. Okay. Trypsin, for instance, one is 0.59 kan, so about 0.5. So in trypsin, one amino acid change will occur every 2 billion years. Does that make sense? For kappa casein, one amino acid change will occur every 3.3 billion years. Okay, right. So based on this, based on the molecular clock, what is the actually the functional properties of a molecular clock? If a molecular clock of a particular sequence is known, we are able to sort of derive ancestry or distance between those two sequences. Contoh, let's say for instance, let's take serum albumin, and let's say that the molecular clock is 2 times 10 power of 9, or 2 times 1 billion years. Let's say, for instance, I do a pairwise alignment between serum albumin in human beings versus serum albumin in cats, right? And when I do an MSA, a pairwise alignment, I find four amino acid changes, right? So how far is the distance in terms of years between serum albumin in human being and serum albumin in cats? Berapa? Anyone can answer? Tahu tak? Ke nak panggil nama ni? Boleh je panggil nama sebenarnya, tapi sekarang korang takut yang tengah tidur. Tahu? Anyone can venture a guess? Let me repeat. So let's say, for instance, when I do a pairwise alignment between serum albumin human being and serum albumin cat, and I find 4 changes, how far is the distance in terms of phylogeny between serum albumin in human beings and cat? If I find 4. Berapa? Tak tahu. Ni kita tanya, kita buka. Berapa?
8 billion?
8 billion? Tak. Okay. So if you find 4 changes, you find 4 changes between those two albumins, lactoalbumins, and you know that for every 1 billion years, two changes occur. Maksudnya how many billion years must it take to have 4? So that's basically number of changes bahagi the molecular clock. Wah, so kita lagi best, dia jawab 7.6 million years. Tak. So let's say, for instance, you have 4 amino acid changes, right? 4 changes between human beings and cats. And the molecular clock is 2 changes every 1 billion years. Sorry, one change for every 2 billion years. For 4 of this to happen, then that means, so 1 change is 2 billion years, 2 changes is 4 billion years, 3 changes is 6 billion years, 4 changes is 8 billion years. Nampak tak? So the distance between the the serum albumin of human beings and cats are actually berapa tadi? 4 billion years. Okay. Does that make sense? Alright, let's make another example. So let's say, for instance, let's take fibrinopeptides. Ni 9 9. Let's say you do an the difference between fibrinopeptide A and B. The difference is, let's say, 5 differences. So how far are the fibrinopeptides A and B apart? So remember, there's 5 changes. So every 9 billion years, 1 change. 18 billion years, 2 changes. 27 billion years, 3 changes. 36 billion years, 4 changes. The 5th change occurs after 45 billion years, which means fibrinopeptide A is 45 billion years apart compared to fibrinopeptide B. Does that make sense? Okay, or we can say that the genetic distance between, or the protein distance between those two fibrinopeptides are about 45 billion years. Okay. Okay tak? Okay.
Betul lagi, Doktor.
Alright. Kita ulang balik So the numbers here tell you the molecular clock of a particular protein sequence. The molecular clock tells you how regular change occurs, right? For instance, for fibrinopeptides, the molecular clock for fibrinopeptides is 9 billion years. That means 9.0 times 10 kuasa 9. So ada kata sini molecular clock for proteins, rate of changes per amino acid site per 1 billion years. So for fibrinopeptides, it's every 9 billion years, there is a change, there's an amino acid change. Okay, right? Which also means that every 9 billion years, one sequence akan berubah dalam fibrinopeptide. Let's say, for instance, you do an alignment between fibrinopeptide, dog and cat, and then you find 2 changes. So the question you might ask yourself is, how long does it take for those 2 changes to occur? If you know that every 9 billion years, one amino acid changes. Okay, so kalau if there are 2 differences, that means it takes 9 times 2 lah. So that means 18 billion years for that change to occur. Okay.
Oh,
Okay.
faham.
Faham dia simple sebenarnya, tapi macam di bioinformatik kan, so korang jadi confused. Let's say for instance, let's look at, okay, kita guna cytochrome C, cytochrome C 20 million years. Let's say when you look at cytochrome C between butterfly and fly, kan? Cytochrome C, butterfly and fly, there is a difference of maybe let's say 10 changes, right? So what is the distance between the fly and butterfly cytochrome C? It's obviously 20 times 10, because you have 10 changes. You have 10 changes, and one change occurs every 20 million years. Nampak ni? To the molecular clock there. So 10 times the number of the molecular clock, then you get how far they are apart. So that means cytochrome C, the protein cytochrome C from fly and butterfly, is separated around 200 million years. What does that mean? That means the process of evolution from an ancestor that diverged into butterfly and fly occurred 200 million years ago. Nampak? Okay, kita lukis. Right. Okay, let's say for instance you have fly tadi okay, buat A, lepas tu kita ada B kan. Ya Allah, tulis pakai mousepad sah. Okay, A dengan B kan. And then when you let's say the molecular clock is 20 million years. Cytochrome C untuk A dengan B. And the molecular clock is 20 million years. Let's say between these two, kan, there are let's say 10 differences. Ya Allah. Okay, faham lah differences. Okay, so if you know the molecular clock of cytochrome C, you can now agak-agak how far along. If I ask you, how are they apart from an ancestor? Let's say ancestor dia G, C lah, mana dia. Okay, so by looking at the differences in sequence data and then knowing the molecular clock, dia kata the molecular clock for cytochrome C is 20 million years. So one change every 20 million years. If there are 10 changes between A and B, then A and B is separated by about 20 times 10, which is 200 million years. Or we can say that this part over here, where the ancestor C evolves into A and B, was 200 years ago. 200. Faham tak? Million, ya Allah. Okay, million years ago. Does that make sense?
Doktor, yang 0.22 dekat slide tu apa?
Mana 0.22?
The cytochrome C 0.22.
Okay, so this is actually how many, maksudnya every 1 billion years, 22.22 change. But let's look at this one. Okay, let's assume that cytochrome C, dia punya molecular clock is 20 million years. I'm not sure kenapa dia berubah kat sini. But let's assume that dia punya molecular clock is 20 million years. Oh betul lah, sebab ini million years, ini billion years. So dia bahagi 3. So 1, 2, 3. Okay, maksudnya every 20 million years, satu change occurs. Kalau kat sini 0.22 change every 1 billion years. So lebih kurangnya ni, betul je ni sebenarnya. Does that make sense? Okay tak? Okay ke tak okay?
Tak berapa okey. Kenapa 0.22 mana datang lagi tu?
Okay. Okay, ni jangan tengok. Sebab okay, the thing is, this one don't worry about the number here. Because every single protein dia ada dia punya own number. I think this is just for, I think Dr. Adam letak ni sebab just as an example kot. Ni nombor dia. But what this means is that cytochrome C ni, dia kata kat sini, dia punya molecular clock is 0.22 times 10 billion years. Which means setiap 1 billion years, dia ada 0.22 perubahan. 0.22 change amino acid. Ataupun, kalau saya pindahkan decimal point ke sini kan, setiap 10 billion years, ada 2.2 amino acid yang berubah inside cytochrome C. Does that make sense? So jangan tak cytochrome C lah. Cytochrome C macam pening sebab dia ada point kan. Look at yang nombor bulat ni, macam contohnya fibrinopeptide ni. So 9 kan. So that means it tells you that every 9 billion years ada one change in fibrinopeptide. Okay. Kalau let's say ada soalan ataupun let's say ada macam when you do a pairwise alignment between A and B kan, and dia punya molecular clock is 20 million years. When you do a pairwise, you find 10 differences. Macam mana kita nak tahu how far they are apart? So by looking at the molecular clock, you times aja how many changes that dia ada, and then you have dia punya distance, which is 20 million years. Maksud 200 million years ni maksudnya ialah the process of where an ancestor berubah kepada A and B adalah 200 million years ago. Faham tak? Does that make sense? Boleh saya assume okay lah. Okay, tak apa. Kalau tak faham nanti, hopefully you guys have some grasp of what a molecular clock is. If you are a bit more confused, nanti I'll put up the recording, the record lecture, then you guys can go ahead. But whatever it is, I can always ulang balik this next week. Is that okay?
Okay, Doktor.
Right. Okay, I'll move on. Macam nak buang ni. Annotate. Lepas tu clear. Right. Okay. So ini tadi lah. So this is actually, this slide over here sort of describes or explains what I just mentioned just now lah. Maksudnya, if you know the molecular clock of a particular protein, and then you know the amount of differences between those two proteins, you can actually calculate how far the evolution distance or the evolutionary distance is between those two proteins. Now remember that because molecular clock hypothesis relies on molecular clock for a particular protein, you can only use the molecular clock for orthologs. You cannot use it for paralogs. Contohnya, you nak tengok the difference between alpha globin and beta globin. Those are two paralogs. They are not orthologs. So you can't use the molecular clock because you don't know which molecular clock to use. Because alpha globin has a different molecular clock, beta globin has a different molecular clock. So which one to use? Tak tahu kan? So you can only use it between orthologs. That means if you're looking at cytochrome, only cytochrome C from different organisms. Okay? Right. So far so good? Okay, right. Okay, so I'm going to move into I think the last part for this morning lah. At 10:20 already. Okay, yang ni senang. When we do molecular phylogeny, the final product will be a phylogenetic tree, which looks something like this, benda ni. Okay, so a phylogenetic tree terdiri daripada or is comprised of different components. The first component is what we call a taxa or a taxon. So every single point in a phylogenetic tree is called a taxon. Multiple taxons are called a taxa. That means the plural for taxon is taxa lah. So in this particular tree over here, A, B, C, D, E, F, G, H, I are all considered taxons, right? So every taxon is located at one point in the tree. But A is at this point, B at this point, G at this point, all right? The taxons represent your sequences. It can be your DNA sequence or protein sequence or even organism. So those are considered taxons, okay? Right. A taxon is also known as an operational taxonomic unit, OTU. What it means is it refers to a sequence lah, macam protein sequence ke ataupun DNA sequence or an organism. Okay, that's why it's called an OTU. Multiple taxons ataupun taxons A and F, they are connected by a branch. So yang line between A and F is called a branch. The line between F and B is called a branch. The line between A and G is called a branch as well. Right? So a branch connects between taxons or taxas. Okay? Persimpangan every single branch or in which where a taxon deviates into two branches is called a node. Right, a node, an intersection or terminating point. So mana-mana titik yang branching out, it can branch out into two new taxas or three or more than two taxas. That point is called a node. All right. That means the area, the taxon can be on a node. It can be not on a node. Okay. Right. So far so good. Okay. Branch dah cerita dah, so it's fine. Okay. So the branches, the length of the branch ni kan 1, 2, 1, 2 ni and 6 ni, it can be scaled or it can be unscaled. Contohnya if it's unscaled, the length is usually the same. Dia macam one unit is the same, and the length of those branches mean dia rarely reflect sequence data. That means it relates to just time. That means the further A and I is apart, the longer the branch will be. Maksudnya kalau let's say the distance between I and A is let's say 10 million years, panjang dia macam ni. If it's 20 million years, dia lagi panjang lah. So the length only refers to time. But if the branches are scaled, the length is proportional towards the change. That means let's say between A and I tadi, there are 5 amino acid changes. So the unit dia akan change lah. 2, 1, 2. That means there are 5 units that separates A and I because there's 5 differences. Okay? Right. Okay. All right. So a node, remember we talked about nodes just now. Nodes is the point in which a branch deviates ataupun a taxon deviates. It can be deviating into two new taxons ataupun more than 2 taxons. If one node deviates into 2 sections or 2 taxons, we call it bifurcating. Bi means dua lah. That means dia akan pecah satu, pecah per dua. Bifurcating. If you have one node deviating into more than 2, kita call it multifurcating. There's nothing called trifurcating, tak ada. Dia ada either bi 2 ataupun multi more than 2. Okay, right. So far so good. So multifurcation, it sort of tells you the quality of sequence data that you have. If you have good enough sequence data, you are able to get a lot of bifurcating nodes. Let's say, for instance, you don't have enough sequence data. Remember, when you did MSA and then you use orthologs or very closely related sequences. If there's not enough differences to tell which one comes before what, then you will have a lot of multifurcating internal nodes. That means they branch out pergi banyak-banyak, right? Contohnya kat sini. So if you look at this particular phylogenetic tree, kan, you can see that certain nodes, like this node over here, they branch out into 2 new taxons or two new nodes. So this particular node, we call it bifurcating. But you also find that certain nodes over here, they branch out to, let's say 1, 2, 3, 4, more than 2. So this node over here, we term it as bifurcating. Right? So why do we have bifurcating? Why do we have multifurcating? Let's say, for instance, if you look at this kan, ancestor kat sini, and let's pakai contoh kat sini. Okay, so whatever ancestor that came before human and mouse, kan, the differences between human and mouse is actually human, mouse, and zebra lah. Okay, human, mouse, and zebrafish. 3 ni. The sequence data between human, mouse, and zebrafish is very good. There's a lot of differences, so you are able to say that zebrafish is the one that came zebrafish is actually very different from mouse and human beings. So because you have enough sequence data, you are able to have bifurcating nodes. You know which one comes before which and which one is closer to one another. But let's say if you look at, let's say, mollusk, annelid, semua tok nenek ni, kan? You know that there's not enough sequence data to tell you which one came first or which one came later, because there is this multiplicating node. So the sequence data yang you have, you cannot decide whether ini tergolong dalam kumpulan ini ataupun tak. So because you don't know, you assume semuanya diverge at the same time. That means one ancestor, dia just evolve into all the ancestors kat sini. Because you don't have enough sequence data. Sampai sini baru you have. So, nampak tak? So multiplication comes because there is a failure to resolve the branching. That means you don't have enough data to suggest the deviation of species. So you anggap je semuanya evolve at the same time, right? So the goal of the phylogenetic tree is to have more bifurcating nodes than multiplicating nodes. Okay, right. So inside a tree, you can also group the taxons in what we call a clade or clades lah. Let's say clade lah. All right. A clade refers to a group. So inside this tree, for example, over here, there are a lot of groups. There is the group which is comprised of taxons A, F, and B. So that's one clade. There's also another clade, which is one group, which is C, H, and D. That's one clade. But you also have A, F, and B belonging to a larger clade, which is actually A, F, B, G, H, C, and D. So that's one clade as well. Nampak tak? One bigger clade. Then you have E, duduk sorang-sorang kan? But E actually is inside the larger clade, which is E, D, H, C, B, F, A, and G, and I. This is one clade as well. Okay, right? So far so good. So clade simply means the groups that you can actually find in a phylogenetic tree, right? So clade ni, what does it refer to? So if let's say, for instance, I have a phylogenetic tree for a lot of types of musang dan anjing, foxes and dogs, right? So you can see that the Arctic fox, the kit fox, and all these foxes, they come from the same clade, one clade kat sini. Then you have the jackal and the wolf that coming in one clade, macam tu. So dia macam groups. Dia macam your own family is one clade, and then you have your cousin is in one clade, but the both of you comes from a larger clade, macam tu. Okay, so that's clade. So far so good. Right. Okay. So another thing is a phylogenetic tree can be rooted or unrooted. Rooted means there is a common ancestor that is known, that there's enough sequence data to tell you that there is one ancestor. So if it's root, it looks like this. There is one vertical line at the beginning of the tree, okay, which is the common ancestor for all the taxons here. If there is not enough sequence data to determine ancestry or an ancestor, you can have an unrooted tree. So you have taxons 1, 2, 3, 4, 5, tapi you don't know which one is the earliest. You only know that 5 and 4 is very different from 1, 2, and 3. But you don't know which one came from what ancestor. So you combine macam ni je. You don't know which one. Here we know 1, 2, 3, 4, 5. We know 4 and 5 is different from 1, 2, 3. But they came from the ancestor number 9. So we know. Okay. So rooted we know, unrooted we don't know. Okay. Let's say, for instance, I tell you, okay, I have 1, 2, 3, 4, 5, kan? And I say, okay, can you convert this unrooted tree into a rooted tree? So one way of converting a rooted tree to an unrooted tree is by inclusion of an outgroup. What does this mean? If you, let's say, for instance, you have 1, 2, 3, 4, 5, these are all, let's say, for instance, ortholog proteins. Let's say beta globin. So beta globin 1, 2, 3, 4, and 5. If you want to root this unrooted tree, you need to put in another sequence, but that sequence must be an outgroup ataupun a group that does not relate altogether. Let's say, for instance, myoglobin. So myoglobin is totally different compared to beta globin kan. So when you put myoglobin inside this unrooted tree, you automatically have a root kat sini. Alright, but this root has an ancestor which is hypothetical yang korang anggap. Because you know that 1, 2, 3, 4, 5 is actually beta globin, and 6 is a myoglobin. And you know that myoglobins are completely different from beta globins, and they are indeed separated by an ancestor. So that's how you root an unrooted tree. Does that make sense? Okay. So to root an unrooted tree, we must introduce an outgroup, which means a sequence which is unrelated to the to your taxons kat sini. All right. Okay. 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 So I'm going to stop here. Any questions? Any questions? Apa dia?
Tak ada.
Tak ada. Okay, good. Tak ada good lah, but hopefully it's okay with you guys. I'm going to stop here because my next session is already started. So kalau if you have no questions, what I'm going to do after this is I'm going to put up the recorded lecture for molecular phylogeny. So please go through it, and then we'll meet again next week on Monday petang. Is that okay? Okay.
Okay.
All right. Okay, so with that, I think we end here. So I will see you next week on Monday. Don't worry about practical report. I'll extend the submission until next week. So whoever dah siap and don't want to think about it, you can actually just submit. And those who want to wait can actually submit later on. There's no hurry, at least on my end. Okay, any questions before we leave? Ada? Tak ada? Tak ada? Okay, kalau tak ada, I thank you very much for attending the class. I'm very sorry. I apologize once again for having it online. Maybe it's hard to learn macam ni. Tapi we'll revisit all these things next week. I'll probably make a refresh about 5 or 10 minutes before I move on to the next part. Okay. Okay. Right.
Okay.
Thank you very much. Okay. Thank you very much. Assalamualaikum. Have a good day ahead. All right. Bye.
Thank you.